background to why we're testing for residual solvents, what's currently occurring in the marketplace, then talk about some Headspace and GC principles. Understanding those principles can really determine how we can test for residual solvents. And then finally, what I'd like to talk about is implementing USP 467, the current revision of residual solvent testing into the laboratory. So we will talk in detail about how the method works, and then overall some tips for implementing it into the laboratory to make it successful. If we look at the marketplace, why are we testing for residual solvents? What occurred is in 1994, Congress defined the term dietary supplement in the Dietary Supplement Health and Education Act. By definition, what they said is a dietary supplement is any product taken by mouth that contains a dietary ingredient intended to supplement the diet. So upon that definition, what then happened is the FDA released a final rule on current good manufacturing practices for dietary supplements. By the definition, and then as a requirement for GMP protocols, or CGMP as it's listed, current good manufacturing practices, these now need to be established in our laboratories. Anytime we're preparing a dietary supplement by formulation or by production, we need to do that under current good manufacturing practices. And what those do are establish requirements of testing to ensure the products are made in a consistent and quality manner and are accurately labeled and free from impurities. And I've highlighted this free from impurities because when we look at residual solvent testing, what this really is a way to look for organic volatile impurities, also called residual solvents. So what we will discuss is one method for doing so, USP 467, and how we can implement this impurity analysis into our laboratory. What we also have seen is effective August 24th, 2007, there will be a 36-month phase-in period for companies, depending on size. So, to operate under current good manufacturing practices and to test for residual solvent impurities, most people are turning to the USP 467 as the way to test for them. So that's what we will discuss in this presentation. If we look a little bit closer, just to give some of the historical background, a residual solvent, by definition, by the ICH, or International Conference on Harmonization, is an organic volatile chemical, or impurity, OVI, that is produced in the manufacture of drug substances, excipients, and products. So overall, this definition defines for us what a residual solvent is. It's an organic volatile impurity, OVI, and also where we need to test for it in drug substances, APIs, drug products, and excipients. So anything used to produce or formulate that drug compound, or in this case a dietary supplement, needs to be proven that it does not contain organic volatile impurities or residual solvents. If we look a little bit closer at the ICH guideline on residual solvents, namely Q3C, which is entitled Impurities, Guidelines for Residual Solvents, this is the emphasis or the basis for all the analysis we are doing today. It is defined by this guidance. What it did, mainly, is summarize residual solvents into class and define exposure limits. So they're based in class by toxicity. What we will find in this document are Class 1 solvents should be avoided, Class 2 solvents are solvents to be limited, and Class 3 solvents are solvents with low toxic potential. So Class 1, Class 2, and Class 3 are the classes of solvents based on toxicity. This is really the definition for determining what solvents need to be tested and how they need to be tested. If you haven't read this document, it's a very good idea. It can actually be found at the website listed here, so you can go online and get this documentation. And it does show the background or the definition to what a residual solvent is and how our analysis need to be carried out. Most importantly, if we look at the perspective of what this document did to the marketplace, the ICH was established in 1990, and it's made up of members of both industry and regulation, with the overall goal of trying to harmonize the pharmacopoeias, or trying to find one static guideline that we can then follow to try and look for impurities in products and substances we create. So the main point here is this group created this document, this Q3C, this quality guideline, and from that we created change in the marketplace. We then saw methods being adopted or developed to try and coincide with this guideline. The USP 467, the EU, they adopted this methodology to coincide with this basic guideline created. As a result, what we then saw was these chapters being created and used in the marketplace. And the USP 467, USP 30 NF25, was the first one to really mirror these ICH guidelines, to use that solvent list, and that became effective 7/1/08. So what we're going to talk about is ultimately the current version of the USP chapter on residual solvents, but we will discuss how we can implement that procedure into the laboratory today. I just want to show as a perspective the old USP 467. So prior to mirroring this document, Q3C, the overall test for residual solvents by 467 contained these 5 compounds and was a headspace injection or liquid injection. There were method options. But overall, we see that this is a very simplistic method. When we look at what's occurring today, what we have to do in our laboratories, the ICH residual solvents, by definition, are based on class by toxicity, class 1, class 2, and class 3. We see that the class 1s have a concentration limit. These are the most toxic, therefore they're solvents that should be avoided. Class 2 are solvents that have a % daily exposure limit. So overall, what we see are the class 1 and class 2 compounds are the compounds we are testing for in USP 467, because those are the ones that are defined and need to be tested by a specific technique, like gas chromatography. Class 3 compounds do not need to be tested by a specific technique. So this comprises our analyte list for our residual solvent testing, class 1 and class 2 compounds. When you look at the standards themselves, when we do the methods, these are the reference materials being used. So we are actually creating standards based on this analyte list, and we see that we have class 1s and we have class 2s that are separated into 2 mixes, mix A and mix B. So to do USP 467, it actually requires these 3 standards, and they are available. Here are catalog numbers, but I just wanted to mention that these are the ones that we will be testing for in the method. If we look quickly at the chemistry of static headspace, USP 467 is a headspace GC method. So as a result, we are going to produce our standards and our samples in a headspace file, and then take an aliquot from the headspace and transfer that into our GC for separation and detection. But if we look at the process real quickly about static headspace, what we are starting with is a sample phase, and we will discuss this in detail because it's a differentiation in the method. But we are going to produce some solution, and then that solution, through changing temperature and changing time, we are going to see partitioning of our volatile analyte molecules from the sample phase into the gaseous phase or head phase. So headspace. So ultimately what we have is a volume relationship inside this file, and then subsequently a concentration relationship, knowing that we are going to form partitioning of both our diluent and our analytes into this headspace and sample from the headspace. So we are going to make these volatile components partition into the gaseous phase until we reach equilibrium, and then pull up an aliquot, which is a common static headspace injection. Static meaning it's not dynamic. We have a static system produced inside this file. To go over some of the fundamental headspace theory, it's important to note that there is a fundamental relationship that occurs in headspace theory, and I'm just going to mention it briefly. If you would like more detail on this, we have a webinar on headspace theory that really describes in detail how this process works. But overall, the peak area we get is based on the concentration of the analyte in the gaseous phase. So how well we partition. This is where our sample aliquot is taken. And that is very simply based on the initial sample concentration over the sum of these 2 variables, which is partition coefficient and beta value. So we can look at these 2 things as the K value, or partition coefficient, being the overall relationship of concentration, or how our molecules partition into the gaseous phase, and then also the beta value, which is the phase ratio or the volume relationship inside there. So what is the volume relationship with our gaseous phase and our sample phase? These are going to be defined for USP 467, but I just wanted to give an idea of what these terms are and ultimately how this produces our results in headspace chromatography. It's an important concept to remember that headspace is not an exhaustive technique. Some analyte partitions, but the majority of it stays in the sample diluent. So we are dealing with overall sensitivities and ruggedness of method based on our ability to form equilibriums and to partition molecules into this headspace. When we look at why this is important, in terms of standard preparation, it's very important in headspace analysis that the way we prepare our standards is identical to the way we prepare our samples. And the main reason for that is, as we noted, this is based on a vial system, a static vial system that deals with the partitioning of analytes between our two phases. In order to make that consistent, we have to make the standard and sample preparation consistent as well. So in many cases, what we're going to see for static headspace is it's really dependent upon dissolving your sample. Once dissolved, we then look at partitioning those molecules into the headspace. So when dealing with this, in standard preparation, we are going to take aliquots from our standard mixes, 1 milliliter, and then prepare our dilutions from that point on to bring us to the regulatory limit needed for that analyte. So we will take our mix and we will put it inside this headspace vial to actually prepare our standards. What we see is that this brings apart what I would call the first tech tip for this method. Class 1 compounds are not readily soluble in water. So if water is the diluent we are using to create the sample phase, it's very important that when dealing with the class 1 compound in preparation, that standard in particular, we minimize disturbances because those analytes will volatilize from that very quickly. So we could see overall just lower sensitivities. And when implementing this method, one of the hardest things to do is to reach the requirement of the class 1 compounds for sensitivity. So standard preparation is one issue. So first, minimize disturbances, and secondly, in the class 1 preparation step, we do actually dissolve some DMSO in the first standard aliquot, and that's very important to create the solubility needed for the class 1 compounds. So make sure that in that first step, that the standard aliquot is diluted in the dissolved DMSO. So make sure that you get complete solubility prior to moving forward. And then we will prepare class 1, class 2 mix A, and class 2 mix B of these standards. When looking at the approach we take for the sample, the same diluent we use for our standards, whether it be water or DMSO, is the same one we use for our actual test article. And this brings about the first big choice we have to make with USP 467. This method is broken down into two sections, water-soluble and water-insoluble options. Based on the solubility of our sample, whether it's soluble in water or DMSO for the water-insoluble option, we then choose the way we are going to move through this system. So based on solubility of the article, whatever it may be, we then look at that solubility as defining the water-soluble versus water-insoluble option. And the main point here is if we look at this technique, it's often called a solution approach, meaning we're going to take a solid sample and we will prepare a solution from that. So we are going to take our test article, our solid sample, as shown here with analytes inside. We are then going to choose our option. Let's say, for example, we use water, the water-soluble option. What we then have is a solution where we have our analytes, our solvents, in that solution. And you can see how we've performed our sample preparation to create this liquid gas partitioning system, or a headspace. And from this liquid phase, we can then transfer our analytes into the gaseous phase for sample. If we look at the technique of USP 467 for sample prep, what it does is it takes a certain weight of article, weighed accurately into a 25 milliliter volumetric flask, and we dissolve and dilute with water or DMF to volume and mix. So we take this and we prepare our solution, just as we do with our standard preparation. What we then do is transfer an aliquot of that 5 milliliters of the test stock solution into a Headspace vial, add 1 milliliter of water, apply our cap, and then do our testing. So we can see that what we are doing are preparing our standards and our samples to meet the same amount in this vial. And dependent upon if it's class 1, class 2 mix A, or class 2 mix B, these preparations may change, but the overall outcome is in this vial we are going to have our test article for our samples, and for our standards, our reference materials at the regulatory limit inside this file. So we are creating standards that have regulatory limit values, and then the amount of weight of sample that coincides with that limit. So that's ultimately what we're trying to do in preparing our samples and standards. And then we run through the actual procedures for USP 467. It's an important thing to note prior to moving on to the procedure that when dealing with this method, the solution approach to Headspace, it's rather important that we actually create a solution. And this could be very tricky, especially looking at some of the raw materials and products that we're trying to test, that finding solubility may be the toughest thing. And what the USP has actually said is that whenever possible, the test article needs to be dissolved to release residual solvents. And in some cases, they have actually said that the drug products may first need to be pulverized into a fine powder, or overall trying to look at grinding the samples or creating something that's a little bit more applicable to create the diffusion we need or the solubility. So this can be done. The main thought here is that by breaking down the diffusion distance, we can then lessen the diffusion time. So overall, increase release or diffusivity of the solvents into the matrices. Overall, just a point to mention that it is important that we create a solution before we move forward with the analytical testing of USP 467. If we look at the design of the procedure for this analysis, now let's start to look at more of the analytical detail. We can see that in many testing, and I just show this as an example of common environmental testing, that when we deal with this concentration in the gaseous phase, it's sometimes common that what we then do are take standard aliquots taken with increasing sample concentrations and produce a calibration curve. Therefore, when we have an unknown, this is a very common type of analysis. We have an unknown, we plug the area count into the curve, we then get a concentration for that unknown. When we're dealing with USP 467, we're not dealing with a calibration of sorts. What we're dealing with is what's called limit testing. So rather than building a calibration curve, we are simply going to run the standard at the regulatory limit and prove that the system is suitable. So what we're dealing with are system suitability requirements and really running our standard preparations at the regulatory limit. And that's how those mixes, that's why they're generated the way that they are, that's why they're prepared the way that they are, because we're going to run our chromatography at the regulatory limit and then look for peaks above that limit. So ultimately, we're dealing with a limit test, and we need to first prove system suitability, which we will talk about the requirements needed for that. The analytical flowchart for USP 467 is actually quite complex. Because we're dealing with a limit test, what we're going to do are run our analytes in procedure A, and we will discuss in detail each of these procedures. But the first thing we do is run procedure A, and I call this identify, because what we're going to do is look for the identification or the matching of any residual solvent peaks in our test article with our test solutions. If any of those correspond with each other, we then have to move forward in the analysis. So we perform everything listed in procedure A, all those analytical definitions. If we have any solvent peaks present at an area count greater than the corresponding standard, so if the peak retention time matches up and it's above the limit of our standard, we then move forward into procedure B. If not, we pass the test and no further action needs to be done. So as we move forward into procedure B, what I like to call this is confirmation. So we first identify in procedure A, and then we confirm the presence of that residual solvent in procedure B. What that requires is we run the procedure listed in procedure B. So what that does is then use an alternately selective column, and we look for that same residual solvent peak matching up with retention time and area count greater than the corresponding standard. If we see that the result in procedure B matches what we had in procedure A, we then confirm the presence of the analyte and move forward into procedure C. If not, the test article passes and no further action needs to be taken. As we go into procedure C, this is what I call quantify. So we first identify in procedure A, confirm in procedure B, and then go to procedure C for quantification. So we actually determine, based on a single point standard, what is the amount of residual solvent present. So this is ultimately where we generate our quantifiable data from this point on. And we can use either system, procedure A or procedure B, whichever gives the most sound analytical data. So if we look at the method, the first thing I want to mention is another tech tip. Depending on the sample introduction system that you're using, what type of auto sampler, whether it be a syringe injection, a pressurized loop injection, a balanced pressure type system, overall we have to look at introducing our sample into our chromatographic system and making sure that it's done consistently and efficiently. So I want to use the example of a pressurized loop because this does seem to be the most commonly used style of injection technique and the one that was actually used in developing USP 467. What we find is this is very similar to the HPLC injection port, where we're going to fill a loop, we're going to rotate the valve, go through a transfer line into the injector of the GC. So it's important to note that as our analyte bands are moving, our sample is moving into our transfer line, it's going to come into this area of our injection port, where we see that we have, here's our septa at the top, our transfer line is going to come in the side, we have a septum purge, we have an inlet seal, so our gas flow is going to come down, it's going to come up through here, and then go out to split vent, whatever the ratio we choose to run this at. So typically what we're going to find in USP 467 is we are running at very low split rates. And that's something that's in the method. It can vary, but commonly it's going to be 3:1 to 5:1 is what's most commonly being used. As a result, we're going to have very low linear velocity moving through this liner, then moving into the column, of course. So as that transfer of our sample band comes in and it hits this high volume, low linear velocity section of our injection port, we could see that the sample band will then widen. What we have found is, first of all, when dealing with a pressurized loop transfer line type system, the liner choice becomes critical, which we will discuss in a moment. But also, another tech tip for trying to implement this method would be baking out the system. So bake out the transfer line and loop between analytical batches. What that means is increase temperature to try and remove any type of condensation. This is especially true for the water-insoluble article option, where you use DMSO, and is actually noted in an addition to the procedure in the second supplement of USP 32 and F27. So it has been added in this second supplement, and it's a very good idea to make sure that this transfer line is clean of any type of impurities from our diluent or anything from our sample. So baking out the system does become crucial. But let's get back to sample introduction. If we look at a transfer line system, it's a very good idea, rather than using your very typical 4mm ID liner, which is ultimately produced to have expansion volume. If we look at the process of a liquid injection, we inject liquid into a heated injection port. What's going to happen is we're going to get expansion of our sample, of our diluent. Let's say we inject 1 microliter into this 4mm liner. What we then get is sample expansion, vapor expansion. And that's why it's 4 millimeters in diameter. In a headspace analysis, we are not doing that. We're not creating vaporization in the injection port. We're creating that in the sample, in the headspace file. So we typically want to use very narrow bore liner, 1 millimeters. As a result, we have less room for analyte band to be actually spread. So we increase our sensitivity on the early eluting compounds, and we create a higher linear velocity moving through the liner. So we actually keep our analyte bands focused. And if you look at what happens, this is an example of a 1301 under Procedure A, and the first system suitability requirement is we need a resolution value greater than 1 between acetonitrile and dichloromethane. In this example, by going from a 4mm liner, the dashed trace, to a 1mm liner, the solid trace, we can see that we've increased resolution because we've simply increased the efficiency or the overall peak symmetry to actually create that resolution we need. So just as a tech tip, when dealing with a transfer line style injection, it's a very good idea to use a 1mm liner rather than the very common 4mm liner, that Borov liner, internal diameter, used for conventional GC splitless analysis. When we look at Procedure A, let's look at an example of actually using this method. The water-soluble article option is what we're going to look at. So when we go through that, we said, our Procedure A is our way to identify. So what we do is we run, by definition, a G43 column, which is either an RTX 1301 or 624 style column of these particular dimensions. And you can either use a 30-meter 3.2 millimeter 1.8 micron or a 30-meter 5.3 ID 3.0 micron. I typically use the 0.32 millimeters because they're more highly efficient columns. But overall, what I'm going to show here is just an example of the analysis being ran. And in this case, I used a transfer line. So I used a 1mm split liner with a Tecmar HT3 auto sampler. So it's a pressurized loop style injection. And I had found that I did get the injection to work at a 5-to-1 split ratio, but this was rather difficult. I did run some testing, and I found for the Tecmar HT3 linked to the Agilent 6890 that the 3-to-1 split ratio actually produced the best sensitivity. So when dealing with the Class 1 compounds, which I'll show in a moment, it's very important that we run, that we find the split ratio that works for the optimal sensitivities. The flow rate is roughly 35 centimeters per second, and the oven program is listed inside the method. So we basically run this, and we can note that this oven program is mainly an isothermal separation. So it's 40 degrees C for 20 minutes, which is going to count for most of our analysis. When we look at the Class 1 compounds, the system suitability requirement is a signal to noise greater than 3 for mainly carbon tetrachloride. It's the lowest in sensitivity, and signal to noise greater than 5 for all the rest. So what we see here is the Class 1 compounds being tested, noting at the conditions listed how low the sensitivity is for this compound. This is going to be one of the first troubles you may actually find in implementing this method, is creating good sensitivity or signal to noise ratio for this molecule. As a result, this brings about what's very important in that band broadening, choosing the correct liner, because overall signal to noise ratio is simply based on the efficiency of the peak. How tight is that peak in correlation to the noise of the system? So all these things need to be taken into consideration. But as we see here, under those conditions, we did meet this analytical specs needed for system suitability, or really proving that this system is suitable for use. These are the requirements listed inside the method. And we can also see on this low sensitivity why it was so important that we make sure that we get good solubility and minimize excessive disturbances in preparing the Class 1 standard. Any loss in analyte could be significant in actually failing system suitability. If we look at the Class 2 Mix A, which is the next test solution being tested for, there is a major system suitability requirement here, and what that is the resolution of acetonitrile and dichloromethane. And those two things I had listed as, I would say, is probably the hardest thing in implementing these methods, the most difficult things to do. The 1mm liner helped drastically in this situation. If we look at our RTX 624, we can see that we are getting a resolution of 1.35, but that is a tight system requirement, and this is a common problem with conventional 624 type columns. But overall, we can see that we are actually creating the system suitability requirements for this method, and this is ultimately what the chromatography should look like. We also see that here's class 2 mix B on the RTX 624. So what we have is the overall analysis of our test samples. Our test article will then be prepared as described, ran in correlation. If we pass the system suitabilities, we can then move forth into procedure B. Before I'd like to move on, I want to mention that there is a column that we have actually designed for pharmaceutical use, and I want to mention it because it has very good use for USP 467, and that column is actually our RXI 624 SIL-MS. So we've actually produced a column that has a certain type of deactivation, RXI, and a certain type of polymer chemistry, SIL-MS, that it still qualifies under the G43 category. But if I were to recommend a column for the analysis, I would recommend the RXI 624 SIL-MS over the RTX 624. And I will show you reasons why. If we look at the class 1 compounds, here's the selectivity we see for our RXI 624. It's very similar to what we get with the RTX 624. But we do have very nice peak shape overall for this column. When we look at class 2, this is probably the main reason why I would choose this column. We have purposely tweaked the selectivity of this phase to create maximum resolution between acetonitrile and dichloromethane. So the system requirement is much more easily met. In this case, we actually have a resolution value greater than 4. So you will pass system suitability much more easily by going to an RXI 624 SIL-MS rather than the RTX 624. And this is the main reason why I recommend this column, is because we see greater resolution in this portion right here, which is the system suitability requirement. I would also like to point out that because we use RXI technology, which is ultimately a way that we create inertness in our columns, we see that the pyridine shape is extremely nice. So it has really good just neutral deactivation for both acidic compounds and for basic compounds. So the methanol peak looks good, the pyridine peak looks good. Overall, it's well designed for the analysis of USP 467, and really what I would recommend for this analysis. When we compare the two phases, the RTX 624, it certainly does pass the specifications for this method. So if you have one in-house, it will certainly do the method. If I were looking at moving forward with this method, implementing it, I would choose the RXI 624 SIL-MS. Mainly, as I said, it has better selectivity, it has better inertness. If you look at the pyridine shape, certainly that's passable, but it's much better on the RXI. And also, the RXI 624 SIL-MS has much lower bleed, so it's actually a mass spec column. And this is one of the things that it's not necessarily important here in this analysis, but it is important if you want to move this column into other areas of pharmaceutical analysis. You have a bakeout, the max temperature limit is about 310 degrees C, very little bleed, 2 picoamps at 300 degrees C, so it's actually a high thermal stability column as well. I just want to mention, if you compare the RXI 624 SIL-MS even against competitive columns, one of the things you may see is the actual resolution of benzene and 1,2-dichloroethane can become a problem. So in these two competitive columns, we see that they're not getting nearly as good a resolution for those two compounds, which we actually finish off here. Don't know how important this is in honesty, because we are running a procedure B, which does get good resolution for these compounds. But overall, to produce the method, it's much more simply done with the 624 SIL-MS. Most importantly, though, if you look at the acetonitrile and dichloromethane, which is a system suitability requirement, much greater importance. If you look at the resolution, we can see that that resolution is very common in most 624 columns. That's typically what we see, which can cause trouble in implementing this method, which is why we've purposely created much greater selectivity between those two compounds. So it's really amenable for USP 467. And also look at the shape of pyridine. On the RXI 624 SIL-MS, very good peak shape for this basic compound. With other deactivations, you could see much greater peak, maybe even some peak degradation as a result. So overall, we can see that in terms of procedure A, the RXI 624 SIL-MS is definitely the column that I would recommend. When going to procedure B, what we then do is now confirm the analysis. So we look at running procedure B under an alternately selective column, which is a stable wax. And again, you can choose between two different dimensions here. It's a 30 meter 0.32 millimeter ID 0.25 micron thickness, or a 30 meter 0.53 millimeter ID 0.25 micron thickness. I would recommend this particular column because it has a more favorable phase ratio of having a lower internal diameter with the same size film thickness. But again, what I will show you is overall system suitability required for this, what you need to get from the analysis. So here's an example of the analytical conditions. Notice in this case what I did is use a gas-tight syringe injection, so no transfer line. And what I had found is a 2 millimeter split liner, even up to a 4 millimeter split liner, becomes much less critical in terms of band broadening. At a 1:5 or a 1:3 split ratio, the 2 millimeter liners seem to work just fine. So we can see that this is pretty much a good choice for just overall injection port setup when dealing with a gas-tight syringe injection. And we see the conditions listed here, all the very common procedures listed under procedure B. We now see that we have, as I mentioned, class 1, the coelution of peak 2 and 3 is very common in stable wax or wax type columns, polyethylene glycol phases, and the main reason why we use 2 columns because it produces alternate selectivity. So we can see an example. In this case, our system suitability requirement is much more easily met because carbon tetrachloride is co-eluted, so the sensitivity is much greater. Therefore, we don't have to pass that requirement. So we can reach the sensitivity requirement much easier in procedure B. The resolution requirement is actually between peak 7 and peak 8, which is cis-1,2-dichloroethene and acetonitrile, which is easily obtained on a stable wax column and most phases, to be truthful. Overall, we can see that this is ultimately the elution order that we now get. So we see that we are getting alternate selectivity when we compare that to our procedure A. And the main reason why we use 2 columns and how we can now confirm the presence of an analyte, of a residual solvent, by matching up both the retention time and the peak height or overall sensitivity, knowing that this is at the regulatory limit that we're running. Class 2 mix B then looks like this, and we can see that we are producing adequate chromatography. It certainly works for this method. And then overall, that is the test. So if we get a hit on procedure A, meaning we have an analyte that matches up with both retention time and a greater peak area, we can confirm it with procedure B, alternately selective column. If we do that, we now need to go into procedure C, which is quantification. So we take a spiked test solution and the unknown solution, and we use a single point standard that's 1 milliliter of 1 to 20 of the concentration limit spiked into the test solution. So that's how we run this procedure. And then very simply, here's the calculation. Based on the concentration of the unknown residual solvent, we can then determine overall, using the test solution in itself to take care of any type of adsorption or partitioning differences we could see from adding the test article, we then base our concentration on that. So overall, that's the residual solvent concentration in parts per million. And we can see how we ran through procedure A, procedure B, and procedure C. The test that we use here, we actually have all the singles available. So we run that individual standard, whatever we have, and we can use source standards to actually prepare from that. So again, we follow the method with source standards to make the procedure work. So the last thing I'd like to mention is to set up this analysis. I just want to mention here are all the catalog numbers for the test mixes, depending on whatever you see for quantitation. We have all those singles available. So these are the standards required. Here are the choices in columns. Again, I would recommend the RXI 624 SIL MS, the 30 meter 0.32 millimeter 1.8, this one, and the stable wax 30 meter 32 millimeter 0.25. Those 2 columns is what I would recommend for this analysis. And then I just want to mention that ResTech does carry a full line of Headspace essentials, all the way from vials to septa, and even any type of replacement parts for the systems that you're using. So we do offer competitive prices in those regards. So we do have the complete source for residual solvent analysis. I'd like to thank you for listening to the seminar today, and I just want to mention that we do have more technical information available at the website page listed here. So we do have a landing page that does contain a flyer to show overall all the data I had shown you is listed in this flyer. We have an application note that you can download or request that goes into more detail on how the analysis is conducted. We also have a retention time index for all the ICH solvents, so depending on the type of stationary phase you're using, if you're developing your own internal in-house method, it shows the type of selectivity you can get. A static headspace tech guide, which really goes into more detail about explaining the technical aspects of static headspace. And then also, if you're interested in possible training seminars, those are available. So if you have any questions or just comments, or really if you're trying to implement this method and you're running into difficulties, please feel free to contact me. My name again is Rick Lake, and you can contact me at rick.lake@restech.com. So thank you for your attention. Have a very good day, and if you have any problems with implementing this method, please feel free. I would be interested to help out as much as I can. So thank you, and